Hello friends, I am Miss PRAJKTA Patel, Assistant Professor and Head Department of Zoology, Papumia Pimpalgaon, Arts, Commerce and Science College, Pimpalgaon. I welcome you all on 3 days National Workshop on CSIR-UGC-NET-JRF LS preparation in life sciences, organized by Botany for You, an online platform for the students of life sciences. Hope you all will be enjoying this lecture. But before proceed, I must express my sincere gratitude toward Dr. Mani sir and all entire organizing team to give me an opportunity to express and interact with a large number of students. Hope all the students will be benefited by this workshop. So let's start the lecture. Apoptosis or programmed cell death, or it may be a cell suicide. This is a very important topic from CSIR net exam point of view. Many of you get questions from this topic, and if you are well prepared from this topic, you are easily bound to score good marks. So let's start the topic. Many of you think that as it is a bad thing for the cells of your body to die, and in many cases that's true also. It's not good for the cell to die because of an injury. However, it also important that some of our body cells to die, not randomly but carefully or in a controlled way. For example, have you wondered how your fingers are formed? It turned into that cell between your developing fingers were instructed to die and a long ago while you were still an embryo, and if you hadn't done so it would have webbed hand, perhaps just paddle of tissue with no fingers. See, in this figure there is the incomplete differentiation in the two toes is occurred, and why it is done because of the lack of apoptosis. So the cells between your embryonic finger dried in a process called as a apoptosis, a common form of programmed cell death. These cells undergo a suicidal condition when they receive a certain type of signals or the clues from the body. As apoptosis is an energy dependent biochemical mechanism of programmed cell death, it is an genetically programmed process that deliberate suicide by unwanted cells in an organism during embryogenesis means formation of an embryo. Metamorphosis means the striking change occur inside the body morphology of an organism and the tissue turnover. As apoptosis is the normal occurrence or the normal process in which there is an orderly sequence of an event leads to the death of cell. It has been noted that there are 10 to the power 10 to the 10 to the power 11 cells in a human body die every day by apoptosis. As apoptosis is a Greek word which means a dropping off or the falling off. See, there are the two figures, that is a scanning electromicrograph, in which one scanning electromicrograph is a normal cell. See, this is a normal cell which is rounded in shape, and another one is the micrograph of an apoptotic cell which exhibits a many surface bulb. These are the surface bulbs are present on the apoptotic cell that are budded off in the cell. The demise of the cell by apoptosis is marked by the well-defined sequence of morphological changes, and these are the morphological features. and they are firstly the shrinkage of cell takes place and it become rounded off, then cell become more and more dense. After the increasing density, then the detachment of the cell takes place from its neighbor cell. After that, the chromatin material present inside the nucleus, it become more dense and separate into the homogeneous masses, and cell will undergo budding and bulbing. The buds break off to become a apoptotic bodies, and after that the phagocytosis by surrounding cells takes place, and these all are event takes place during the apoptosis. And when phagocytosis occur, after that there is the removal of apoptotic bodies takes place. These are all main events occur in the apoptosis. See the sequence of cellular event occur during the apoptosis. See in this diagram here there are the morphological changes are showing. Firstly, cell round up and the nucleus inside the cell is collapse. The cell become more and more dense, and after that the chromatin material undergo condensation, the cell undergo bulbing and it forms the buds. After that buds break off from the cell, these are the buds are break off from the cells and it form the apoptotic body, and this apoptotic body is then phagocyted by the phagocytic cells. These are all events occur during the apoptosis. And then next is the history. A German scientist Carl Vogert was first to describe the principle of apoptosis in 1842, and in 1972 Kale was the first who introduced and described the term apoptosis in publications, and for his description of apoptosis Kale received a Paul Ehrlich and Ludwig Prize on March, and he shared his prize with Boston biologist H. Robert. A classical studies were performed on C. elegans. C. elegans is the nematode, and in 2002 the Nobel Prize in Medicine was awarded to the Sydney, John, and Horvitz for their work identifying genes that control the apoptosis, and the genes were identified by studies in the nematode C. elegans and homologous of their gene functions in human to regulate the apoptosis. So many of us have questioned that why cell commit suicide, and what is needed for the cell to suicide. So the reason is that programmed cell death is needed for proper normal development as mitosis. For example, the resorption of a tadpole tail in frog, the formation of fingers and toes of fetus required for the removal by apoptosis, then the formation of proper connections, that is synapses between the neurons and the brains. So see in this diagram there is firstly there is the metamorphosis occur inside the frog, in which there is the resorption of the tail takes place. In adult there is no in adult frog there is no tail, while there is the also removal of an cells between the finger takes place, and this process is called as a apoptosis. So programmed cell death is also needed to destroy the represent threats to the integrity of an organism. Uh, when the cell infected with viruses, cell with a damaged DNA, or a cell which having uncontrolled division, that is the cancerous cell. When these conditions are developed inside the body, then a cell undergo a programmed cell death. When in which situation the apoptosis is occur, there are two situations. That is, one is the physiologic situation and another is the pathologic situation. At that time, apoptosis is occur. So, in physiologic condition such as during embryogenesis or involution of hormone-dependent tissue, cell loss in proliferating cell population, elimination of harmful self-reactive lymphocyte, death of host cell. At that time, the cell needed to die. And in pathologic situations such as when DNA is damaged, accumulation of misfolded proteins, cell death in certain infections, and pathological atrophy in parenchymal organs are the main reasons for the apoptosis. See, in this diagram, there is the morphogenesis occur. Morphogenesis means that there is the elimination of excessive cells takes place, and this elimination occur by the process of apoptosis. See here also there is the some non-functional cells are there for 1, 2 and 3. These all non-functional cells get died by the process of apoptosis. Here also there is the elimination of some self antigen recognizing cell. These pink colored cells are the self antigen recognizing cells, and during embryogenesis these cells are removed or eliminated by the process of apoptosis. Similarly, the excess cell are removed by the process of an apoptosis during the embryogenesis. This is the overview or of the apoptotic pathway, which shows the main pathways and their concluding way for the apoptosis. So, the initiation of apoptosis is tightly regulated by activation mechanism, because once the apoptosis has begun, it leads to the death of cell. There are the three main apoptotic pathways. First is the extrinsic pathway. It is also called as a death receptor pathway. Another is the intrinsic pathway. It is also called as a mitochondrial pathway. And third is the perforin granzyme pathway. The two best understood pathways are the extrinsic pathway and the intrinsic pathways. The intrinsic pathway is activated by the intracellular signal uh, generated when cells are stressed and depend on the release of protein from the intermembrane space of mitochondria. That is, it is then extrinsic pathway. It is activated by the extracellular ligand binding to the cell surface of the death receptor. This is the death ligand and this is death receptor. When extracellular signal bind to the receptor at that time, the extrinsic pathway is leads to the apoptosis. And third is the perforin granzyme pathway. In the perforin granzyme pathway, there are the two main components, that is granzyme B and granzyme A. These are two important molecules which induces apoptosis via the DNA cleavage and the activation of the caspase 10. See in this diagram, once all three pathway, firstly there are the initiator or the signaling caspases get activated, that is caspase 8 in extrinsic pathway, caspase 9 in intrinsic pathway, and caspase 3 in perforin granzyme pathway. All these are the initiator caspases are activated firstly, and then it activates to the executional path caspase, that is caspase 3. Caspase 3 is the effector or the executional caspase, and once this caspase 3 is activated, then it leads to the degradation of an endonuclease, degradation of chromosomal DNA, then protease activation, which leads to the breakdown of protein present inside the cell, then degradation of nuclear and cytoskeleton proteins such as tubulin, myosin, actin, cytoskeleton breakdown are occur, and after that there are the formation of an apoptotic bodies takes place. When the apoptotic bodies are formed, it expresses ligand for the phagocytic cell receptor, and finally the apoptotic bodies are uptake by the phagocytic cell. And this is the overview of all apoptotic pathway, that is extrinsic pathway, intrinsic pathway, and perforin granzyme pathway. These are the pathways. Then next is the caspases. Uh, Caspases is the common component in all the three pathways. Caspases means the cis-dependent aspartate specific protease. The name so because it contains a key cysteine residues in a catalytic site, and it is selectively cleave all the proteins at site just C-terminal to the aspartate residues. The Caspases is the family of intracellular cysteine protease that play an important role in initiation and the execution of apoptosis. All are synthesized as inactive procaspases. Inactive procaspases also called as a zymogen because they are present in inactive forms. When this zymogen is splits into the large and small subunit, and it forms the heterodimer, and these two heterodimer assembles and form the active tetramer. To the date, there are more than 12 caspases have been identified, and these are broadly categorized into the signaling or initiator caspases, then effector or executional caspases, and third is the inflammatory caspases. Firstly, we will see the signaling or the initiator caspases. These are the caspases which initiate the process of apoptosis, and they may include caspase 2, caspase 8, caspase 9, and caspase 10. These are the signaling or the initiator caspases. Then next is the effector or the executional caspases. These caspases coordinate the execution phase of apoptosis by cleaving multiple structural and the repair proteins, and these are include caspase 3, caspase 6, and caspase 7. And next is the inflammatory caspases. These are involved in the inflammation of the cells, and they include caspase 1, caspase 4, and caspase 5. There are many other caspases are also identified. It include 11, 12, 13, and 14 caspases. And in in all three caspases, there are mainly important caspase is the caspase 3, that is CPP32, which play important role in the cascade of apoptotic event. The target of caspases is to degrade the inhibitor of DNase, and it leads to the activation of DNase, and because of the activation DNA undergo degradation. Caspases also degrade the nuclear lamins, disruption of nuclear envelope, actin, tubulin, and myosin, and it also degrades the protein present inside the Golgi bodies. So, Caspases work like this. So let's see the extrinsic pathway in deep, how the pathway is continue. Firstly, there are the main four components are present in the extrinsic pathway. These are date receptor, date ligand, adapter proteins, and next is the caspases. The as I firstly already told you that extrinsic pathways are the activated by the extracellular ligand binding to the cell surface of the date receptor, and which leads to the formation of the date inducing signaling complex. So date receptor and their ligand. The receptor are those who receive signals, and date receptor are belong to family TNF, that is tumor necrosis factor, and these receptor have date domain which play a critical role in transmitting the date signal. So the best categorized receptor and ligand corresponding to the date are include the receptor is fast R, which having corresponding ligand fast cell, the receptor TNF R1, which having corresponding ligand TNF alpha. These are the and so many on receptor and ligand present uh, on this cell. So see in this diagram, this is the date inducing signaling complex. Here T and FR1 receptor. It is present on the responding cell, and this is TNF alpha. It is a ligand or the signal, external signal. When it bind to the receptor, then activation of the date domain takes place. Here TRAF2 is bind to the date domain present on the receptor tumor necrosis factor, and it is again associated with the date domains such as TRADD and FADD, and when it become active it convert procaspase 8 into the active caspase 8, and apoptotic pathways continue. That is TNF alpha is the ligand, TNF R1 receptor present on the responding cell. This is date domain, TRADD, FADD, TRAF2, and procaspase 8. These are the components of date inducing signaling complex. So next we will see the main event occur inside the extrinsic pathway. Firstly, there is the binding is occurred. After that, trimerization of the ligand receptor occur, then the recruitment of date domain occur. After that there is the formation of an date inducing signaling complex, then activation of caspase, activation of effector caspases, and lastly the apoptosis is occurred. So see in this diagram, firstly what happen when in extrinsic pathway, when date ligand, that is TNF alpha, bind to the receptor, that is date receptor, that is TNF R1 receptor, then the date domain get activated, and after the activation it convert procaspase 8 into the caspase 8, and then this caspase 8, after the auto activation it convert procaspase 3 6 7 into the caspase 3 6 7. In this same way the caspase 8 also inhibit the BCL2, due to which bad form the pore on mitochondrial membrane, and because of pore formation cytochrome C, this is the cytochrome C, it is released outside the mitochondria, and it lead to the death of cell. So also caspase 8 activated caspase 3, and it also inhibits the BCL2, due to which there is the activation of bit occur and the date of cell takes place. This is all about the extrinsic pathway. The signal is external here. Then next is the intrinsic pathway. The intrinsic In the intrinsic pathway, there is the intracellular signal is present for the apoptosis, and the main component in the intrinsic pathway is the BCL2 family protein, cytochrome C, adapter proteins, and caspases. So first is the intrinsic pathway and BCL2 family. The control and the regulation of apoptotic mitochondrial event occur through the member of BCL2 family of proteins, and there are the two proteins are include. One is anti-apoptic protein, which inhibit the apoptosis. These are BCL2, BCLX, BCLXL, and BCLW. And another protein is the pro-apoptotic proteins, which promote the apoptosis. These are BAX, BAC, BEAT, BAD, BEAM, and BEAK. And the main mechanism of action of BCL2 proteins family is the regulation of the cytochrome C release from the mitochondria via the alternation of the membrane permeability. Means anti-apoptotic proteins which inhibit apoptosis, and pro-apoptotic protein which promote the process of apoptosis. So let's see the intrinsic pathway. What happen in intrinsic pathway? This is the nucleus. This is cell. When cells subject to the high energy radiation, the DNA get damaged. Here there is the damaged DNA. When cells subject to the high DNA radiation, high radiation, at that time, firstly cell start to repair itself, but if cell fails, then it undergo apoptosis. The tropic factor, this is red colored, is the tropic factor, and this is tropic factor receptor. When trophic factor, this is the signal or the ligand, when bind to the trophic factor receptor, at that time phosphorylation takes place and it activate inositol triphosphate. After the activation of the PI3, that is inositol triphosphate, it activate protein kinase B. Then phosphorylation of the BAT takes place, and this BAT inhibit the BCL2 protein. If DNA is damaged, then it produces protein, that is PUMA, and then it travel from nucleus to the cytoplasm, and it also inhibit the BCL2 proteins. Due to the inhibition of an BCL2 polymer, it forms the mitochondrial membrane and produces pore. See, this is the uh, mitochondrial pore. Due to the pore formation, there is the cytochrome C present inside the mitochondria, and this cytochrome C is moves outside, and then it forms complex with APAF1 and procaspase 9. Because of the formation of an complex, procaspase 9 is converted into the caspase 9, and then caspase 9 is convert procaspase 3 to the caspase 3, that is main executional caspase. And after that activation of an caspase 3, then it bring about the killing of many molecules. So ultimately the cell undergo death. this is all about the intrinsic pathway. Then next is the malfunctioning of an apoptosis. Malfunctioning means when cell undergo abnormal process at that time, means when apoptosis occur in a very small amount, this is called as a inadequate apoptosis, and when apoptosis occur in large amount, it is called the extreme apoptosis. So when apoptosis occur in small amount at that time, it may lead to the cancer may be colorectal, hepatic, prostatic, leukemias or neuroblastoma. It may also develop a autoimmune disorder such as systemic lupus erythematosus and myasthenia gravis. It may also causes the frequent infection of viruses. But when apoptosis occur at extreme level, means cell die uh, in extreme level, at that time it generated the neurodegenerative disorder or the diseases. It may be Alzheimer, Parkinson's, Huntington, uh, spinal cord injury, etc. It also leads to the cardiovascular diseases in which heart may be failure, myocardial infarction, or strokes are generated. It also leads to the formation of an hematological diseases include a plastic anemia, various syndromes. The other disorder also develop which include inflammation, sepsis, AIDS, polycystic in kidney and etc. So next is the don't confuse with in necrotic cells and the apoptotic cell. As we already learned about the apoptotic cell, that is, it is the normal process of the cell suicide, but necrosis is the accidental death. Here, in both the processes, the cell undergo death, but in necrosis, there are some morphological changes are different than the apoptotic cell. Here, what happened? Here is the enlargement of an volume, then swelling of cytoplasm and mitochondria takes place. Then it lost the membrane integrity. Uh, and there is no vesicle formation occur in the necrosis, while in apoptosis firstly the volume get reduced, the shrinkage of cytoplasm takes place, no loss of membrane integrity, then formation of apoptotic bodies takes place, and then the condensation of chromatin and DNA fragmentation is occur. So here the first image is the process of necrosis, and this is the process of apoptosis. See, these are the two normal cell. When this cell undergo necrosis, here firstly the cell undergoes swelling, then there is the mitochondrial morphological changes takes place, pattern of chromatins are conserved, and then there is the loss of membrane integrity, means this integration of an membrane takes place, while in apoptosis there is the condensation or the bubbling or the budding formation takes place in the cell. These are the fragmentation and the buds are formed, and then there is the formation of apoptotic bodies takes place, and this apoptotic body is then engulfed by the phagocytic cell, and this process is called as apoptosis, and this is called as a necrosis.